When we look at a chemical reaction, the enzyme name usually appears above or below the reaction arrow because it's not participating in the reaction, it's just facilitating it. So let's look at this particular chemical reaction, which is the phosphorylation of glucose, which means we're adding a phosphate group to glucose. And this is the first step in a process known as glycolysis. So these P's with circles around them represent phosphate groups, and they're going to come a molecule of ATP. Now, glucose is known as the substrate. It fits inside hexokinase, and it is a reactant. So a substrate is always a reactant. And it fits into what's known as the active site of the enzyme. And notice these curly ribbons all around are the protein backbone of the enzyme engaging in secondary structure. And this active site is lined with the side chains of different amino acids which have attractive forces and a particular fit for the D-glucose molecule. And this is known as substrate specificity. Most enzymes only are specific to a few molecules and some only to one molecule. For example, L-glucose or even D-galactose, which only varies by one chiral carbon from glucose, could fit into this spot. So hexokinase specifically fits D-glucose and allows it to obtain a phosphate group from ATP. And as we think of glucose fitting into hexokinase, it is probably a good time to look at the two different enzyme substrate models that you'll be responsible for. So for an enzyme to work, it has to interact with the substrate. So remember, the substrate is the reactant, and the first model we'll look at, the older one, is known as a lock and key model. And in the lock and key model, the active site of an enzyme is a rigid, inflexible shape, sort of like a key, sort of like a lock, and it's an exact complement to the substrate, which would be the key, and they fit together just like a lock and a key would. The induced fit model states that the enzyme's active site is actually flexible and roughly complementary to the enzyme, and sort of changes its structure as the enzyme and the substrate interact. And if we look at this structural graphic of glucose interacting with hexokinase, we see that hexokinase actually changes its structure when it engages in the enzyme substrate complex, which means that glucose and hexokinase are actually interacting with one another. So there's much more favor for the induced fit model because it seems to be more aligned with the evidence that we have of how enzymes and substrates interact.